biochemistry or organic chemistry. Right? So my feeling here is Creighton is very good at lecturing and give nice talks, very attractive. And the poor professor give the hard subject here. And tell you the real story, I opened the RNA biology course in my institution this year, and I got 0 enrollment. I couldn't make it for this semester. So, but I hope I convinced you this is important. And the reason I came to the field and work on it is my advisor, Dale Dean, and it's 10 years. He passed away. And this April, at Ohio State, where I started getting my PhD, we have a memorial workshop, symposium organized by the department. And many former trainees here. So this is Biao's long-term collaborator, Dr. Li Yi from China, and very prominent plant virologist in China, published in Nature paper this year talking about receptor or what kind of regulation. And this is his postdoc, Yijun Qi, and many of our generation read his paper, how single nucleotide change the symptom, make a lethal strand, how 5 mutations makes the viroid move out of the bundle sheath to enter mesophyll cell, different boundary of the leaves. And many pioneering work. And this is Xue Hua. She's kind of senior in me, not only in Biao's lab, but also in my college in China. Okay, when I was undergraduate student working in a lab, she was a master student there. My bench was like next to her, so I knew her.
What's her name?
Xue Hua Zhong. So she published several papers about how RNA motifs like loop E, those base pairing, everything, how important to keep the function, to keep the structure. Okay, and this is Asuka Itai. I just mentioned the very first 2 papers talking about RNA silencing triggered by the viroid. Okay, and this is the so we all gave talks, and this is the lab we used to work at Ohio State. And when they are pretending we're still working there, take a picture. At least 2 benches. So that was my bench, that was Xue Hua's bench, and before me was Yijun's bench. So at least 3 of us work in that space. And I worked there for 12 years, 11 years or 12 years. So, and this is the seminar and a lot of people. And they are the virologist, plant virologist, Q Feng. He worked in Dr. Resner's lab for viroid. And he told me, if you do PCR, clone DNA, clone viroid, and use that PCR to infect tomato, you can infect it. That was very early in the 1980s or early 90s, the trick. So a lot of viroid research there, and this is Biao's wife and Biao's son, and we're happy there. So after 10 years, we have the chance to get together. But just to let you know, the subject I explained to people, oh, you work on RNA, oh, RNA structure. Great. That's it. But this is exciting, fundamental, important. And Biao was trained as a taxonomist in the forestry department, recognizing trees, grasses, flowers, has no molecular background at all. His PhD, his master, and then PhD at Cornell University, look at the microscope, electron microscope, dedicated in those like microstructures, particularly plasmatasmata. And when he looked for a postdoc job, okay, UC Davis, Bill Lucas lab needs someone to look at those how virus move from one cell to another cell. And Bill thought, great opportunity for me to learn molecular biology. That was in 1994. And Bill said, no, just look at the microscope. Okay, for another 3 years, look at how protein and RNA move between cells. But he learned a little bit of molecular biology. And in 1997, Bill was looking for a job. And that time, the US job market was terrible. There was 2 plant openings, one is at Oklahoma State. And they're looking for anatomist looking at the plant structure. anatomy, plant anatomy, to teach that course. And Bill had that expertise. So fit into that position, and he got a job. And 1997, 1998, he had a very small lab space, 1 or 2 students, including Oscar, and worrying about what to do. He cannot compete with Bill Lucas. And whatever, he tried to propose several times and then was told, yeah, Bill is great, you cannot do the same thing, you cannot get funded. He started to look at viroids, and he was introduced to Bob Owens by Rick Nelson, a plant biologist. At that time at Noble Foundation in Oklahoma, like 1 hour driving away. So they worked together, starting working on viroid. And Bill said, it was fascinating trafficking RNA. He had no clue about RNA. He bought a kit, transcribed it, failed, didn't get anything. And now the message I want to deliver is, he had no background on RNA, didn't understand anything about it. And he was so brave, jumping into the area, working on a structure, of course with help with other people, okay? And it turned out such a great career. And the people here, Yijun, after PhD, postdoc training in the lab, moved on to RNA sequencing lab. And he was the first to know how agonal protein sort different small RNA. The first nucleotide matters. He was the correspondent also of that Cell paper, published in 2008. And Xuehua now work on the epigenetics, which is perfect because epigenetic, the RNA-dependent DNA modification, was identified, discovered using a viroid system. Okay, and I'm the only one from the lab at that time. I was a postdoc and still working on viroid. And the thing why I'm not continuing on that viroid structure thing, it's a bit complicated, because Bill had another PhD student in the second year, Jian Wu, if you know, he published some viroid paper. And I was told I cannot sabotage his PhD thesis, so I was not allowed to work on some of the things. They relied on a collaborator, Leontis, Nikos Leontis. But Nikos Leontis unfortunately passed away in 2020 in a tragedy car accident. But we can still do some of the work, and sometime I will pick it up. And that's why I'm here explaining the RNA structure from the basis. And I don't expect you to learn it and think about it like a programmer. You have the cheat code, and you do it, and you need to practice with real data. But you will be there, okay? And RNA structure, like the coding, and in the beginning you feel a little bit, you need a little bit jump start. But once you master it, it's something so unique, and you can do things beyond viroid. I'll give you one example in the end, okay? So I can switch to.
You can use your pointer to switch. There's left and right, yeah.
No, I'm switching to different slides. So that was it. Yeah, sorry. Because in the morning, that slide, I don't have. Yeah. Now I'll come back, act like an organic chemistry professor or biochemistry professor, doing something challenging, but let's go through it. No. Okay, so we start from the building block of RNA. So here you can see this is a nucleotide. It has a base, and it has a sugar, 5 carbon sugar, and this is monophosphate group. Okay. So far so good, easy, right? You saw it in the chemistry or organic biochemistry textbook, and the reason I show it is this part is joined together with the phosphate group and this oxygen hydroxyl group joined together to make the backbone, so you can have a long chain of RNA. So from one Lego piece to join together, you have a long one. And what makes it unique here is this base. And sugar is also important, but base here, they really distinguish each one. So if you want to see something different, you cannot tell the difference by this part, and it's this part that makes each unit different, okay? And why they draw old N in blue here is because N, they have the capacity to attach to other things. They're sticky. So that makes them reactive. So if things want to come and interact with this base or recognize them, that's oftentimes give them a hand, sticky hand, so they can do that. Okay, so this is a building block of RNA from one piece to join together, and you have different block, just 4. Okay, not too complicated. And when we talk about base pairing, we always say A pair with U, G pair with C, and the pairing is based on hydrogen bond. Okay, A and U has 2 hydrogen bonds, it's a little bit weak. G and C has 3 hydrogen bonds, a little bit stronger. Okay, that's most cases for DNA. In DNA, it's always Watson-Crick, this base pairing. What is Watson-Crick? You see this in this nucleotide adenosine. I draw out this WC is Watson-Crick. So this part, they call it H, or this front, okay, is called Watson-Crick front. And this is the Hooksting, another one. So this part, so different angle. And this one is a sugar, so you connect to the sugar part. In RNA, all 3 can be used for base pairing. Now, if you play a game, now you see the rules become complicated. Instead of simple Watson-Crick rule, now you have different diversity now. Okay? So different age can be used for interaction. And each nucleotide all has these 3 ages, can be drawn like this. Okay? Still remember the rules for the game? Yeah. So from base pair, sticky end from N, mainly from this N, you can see the age for interaction, either hydroxy group or the N, they are involved in hydrogen bonding. Okay? Now, when you have base pairing, what it means is through the hydrogen bond, mostly from the N, stick out to other part, if they pair, like say Watson-Crick, from this carbon, the number 1 prime carbon position in the sugar to this sugar, there's a distance. You think the 3D space, there's a distance. You can measure distance if you have a ruler. Okay? But you don't have to do it because people have measured all this distance in the crystal structure and the NMR structure. They summarize it already. Okay? So I will show you where they deposit all the information there. Okay? If the distance is the same, and this branching out, you see this kind of base sticky and has opposite position. They are trans orientation, or they have the same orientation, that's cis orientation. If they have the same orientation, for example, both are trans, and they have the same distance from carbon prime 1 prime to carbon 1 prime, they can replace each other. Not always, but often. You think about it, it's like you have a Lego piece, you have a green, red. They occupy this space. You take them out, you use yellow and blue. Still take the same space. Make sense? So they can be substituted as long as they occupy the same space. And the same space are measured by this. Okay? Things can get a little bit complicated because you learned the last talk in the first session today in the morning, this methylation or other modification. And how would that affect it? For example, we still see Hooksting age and this. But if you, for example, here you have M6A modification. Okay, the nitrogen originally is active. They can form hydrogen bond with others, but you blocked it with a methyl group. Now they are not active anymore. So modification can affect RNA structure or RNA protein interaction because that hydrogen bonding is being disrupted. So now I'm going deeper and deeper So we have building block. We explain the 3 ages. They all have 3 ages. And the key thing is, can they form the hydrogen bond or not? Okay, you can rotate it, play with it, as long as they occupy the same space. Like LPC, you can try and see them fitting or not. And we have a long-time collaborator when I was at Ohio State. It was from 2004 to until he passed away in 2020. We have like 12, sometimes, so 16 around that time. Yeah. And what Dr. Leontis does is he look at into all the base pairing in the crystal structure and NMR structure and subtract all the information and summarize in this database. You can Google it, RNA base pair catalog. And it has all the orientation and distance, so carbon 1 distance, everything. And it's summarized in this kind of table. And you see the big red cross means A and A, they cannot form the cis-Watson-Crick-Hughes interaction. In all the database so far, this has never been found. Suggests they cannot form in nature. So it means the A cannot use its Watson-Crick age to interact with another A's Hughes age. It's just not possible. But a C can do it with a C. So you see this cross. And there are 12 different combinations, and each combination is summarized a table. And how do we know each one can substitute another one? You look at here. In the middle, if it's blue, means they have the same distance or the closest. And that's the summary in that. Okay, so CC, of course, and it can maybe be replaced by UU because the variation is small enough. Okay, some of it just cannot form. And you look at these 2 tables, you immediately will know how to design mutant. For example, if I see a structure like that, I want to disrupt it. You go to the RNA base catalog and find the one that can replace it or can disrupt it. So you have function capped mutant and function disrupt mutant. And the beauty about the viroid is you can generate a mutant easily, and in fact plants, you can have the outcome, very clean outcome, transmission or not, replication or not. If you find some structure in ribosome RNA, you just couldn't do it because they are essential in the cell. But viroids is not essential for plants. You can put into plants and test the function. Okay, by that, viroid is a very great model to test RNA structure and function, all these things. So this is the basis. So it sounds very complicated, but when you break it down to each steps, it's not that hard. Okay. Now we have this in hand, and we want to go a little bit. It's all base pairing if you look at 1-on-1 pairing. And at the larger molecule level, the RNA structure can be broken down to different levels. The primary is the main sequence of RNA, and the secondary likely is the free energy driven, forming these stems at the loops. Okay, the stem is up base pairing, the loops is still base pairing. It's just not Watson-Crick. It can be other base pairing, like Hughes Watson-Crick. And the program cannot predict it, so they give you a bubble there. It doesn't mean it's empty hole. It has different base pairing in those loops, and has been well demonstrated in multiple viral loops and in multiple other RNAs. Okay, so that's why it's all drawing different pairing here. It means they are not just free, empty, like flowing around base pairs. They are highly arrayed, highly arranged base pairs. And then with that, 3D like a molecular interaction-driven information give you a more higher order of structure. It can be small molecules, it can be magnesium, it can be a water molecule. Some examples show that. It can be protein, like chaperone or other protein bound to it, give you the structure. And then you can have higher order, like in the ribosome, sometimes they form a kissing loop So it means two different loop structure, long distance, and come together. So we love them kissing loop because they touch. Okay. And if you look at the 2D presentation of RNA, this is tRNA, like that. In reality, it's like a clover. It's really twisted. Okay. You can, if you want, contact me. I can. This color paper, scissor paper artwork. So you can cut it out, stick it, then you can get a tRNA shape, and you will know how it from the primary sequence to a higher order, and it really looks different. Okay. Now, based on that, another thing is RNA and DNA are very different. We all know, like, I think this is like a double helix when they're pale. Like Quinton just said, we should consider viroid as a double-strand thing. If it's double-stranded, it's like a rotating spindle, like this, like a helix shape, right? But DNA helix and RNA helix are different. Even when they are perfectly matched double-strand, they are still different. This is A form of RNA and B form of DNA. You can see the twisting is more even in DNA. Minor groove and a major groove is more open, and this minor here. But the RNA somehow cannot form that perfect ladder shape like you see some of like architecture ladders. You can very nice spindle like that. It somehow spin and then move out of a little bit. So it's here. This bunch out. It's not perfect. Just one cylinder view of it. So because of the angle, everything is different. Okay. So when you have a long stretch of double-strand DNA, it forms this kind of really nice spindle. But when you have a long stretch of RNA, it's kind of awry and not even. Okay, so keep that in mind. Based on that, so this is one of the Xuehua's paper published in 2008. What she does is actually very simple. Using potato spindle tubal viroid, and it has 27 loops. The loops means in the secondary structure, they don't draw like a complete complementation. She introduced mutation to close them, become double strand. So she called loop closed mutant. And just put them back either in the protoplast, see if they replicate, or put them into plants to see if they move from one leaf to another leaf, trafficking. Okay. By doing that, you can screen all 27 loops, whether they are involved in transmission, movement, or replication. Okay? And the blue ladder labeled with R means they are involved or important for replication. The ladder labeled with T are important for systemic trafficking. Okay? So some of the loops are difficult to tell. The reason is when you mutate them, often give you mutation when you put it back to plants. So you don't know if new mutation does the job or what. So it's hard to conclude. But most others, based on this screening, they show which are the replication important loops, which are the trafficking important loops. Okay? But RNA cannot does the job by itself. It takes two to tango, right? So it needs oftentimes a protein factor. Okay? And you can imagine the loops are mostly look different because it's like different bus stop or whatever. They have sign, stick out. Okay, you can interact with me. I can have this function. Interact with another one. They have different function. So they have different shape, different composition, and used for different function. You can think this way. When it comes to the loops, give you one example, loop 19. This is one I did most recently with another PhD student the same time as me. After my advisor passed away, and we said, okay, let's work out this paper and send it out. So we use the prediction, like what I just told you, in the RNA catalog RNA-based catalog, we design mutants and see what's the best explanation of this simple loop called loop 19. And we found this cis sugar and sugar age interaction. So each base use their sugar age to interact with each other. Okay, and this is the circle, different symbol to draw it, so you don't have to write like sugar here. So then you know this is sugar, and this is cis in these solid shapes, and empty as trans. Okay, so it's all symbolized. It's like a cheat sheet. If you don't know, you just print it out and help you to look at it. After 2 hours, you don't need it. Okay, take some practice. But it's not that hard. Now, once you know all this, like when I was at Bial's lab, everyone is screening different loops and look for their function. I was the outlier. Bial sent me, you need to look for the host factor. Okay, so I was left alone to work on different proteins. And so when I look at how protein RNA interaction, and this very nice summary, so protein and RNA, their interaction is not random, and not like DNA. DNA is more like transcription factor recognized DNA promoter region, for example. It's base, different like a sequence, like 6 or 8 specific sequence or consensus sequence, you know that. But RNA is not. It's really structure-based, and that structure gives, for example, here, an RNA base, and stick it out. So the red is the red is RNA, the blue is the protein. So like this base is sticked out and become a unique site, so the protein can recognize it and bind it. Okay. And in old RNA protein interaction in the database, about 1/3 use this mode to interact. Okay. And RNA backbone, in this case, there's no specificity. They just generally bind RNA, like double-strand RNA, as long as double-strand RNA, they bind. And in this case, account for about 41% RNA protein interaction. And this type of interaction has very less sequence-dependent specificity. And this is more sequence-dependent specificity. Another is RNA sugar to protein interactions. And for example, when you do the helix, somehow you have one region stick out a little bit. They are not standing in the line, like a bad student. And that part can be recognized by protein. And that accounts for about 23%. So this is the basis how RNA and protein interaction. So I will talk so much to give you an idea why we look at loops, why we don't look at double strand, because double strand is rarely to have specificity, right? And the specificity is random only in some cases when this kind of doing that helix structure is extrude out a little bit, but that's not base-specific specificity. Any base at that position can be recognized, okay? And it's not because of the backbone. If it's double-strand backbone, they attach to that. There's no specificity. The specificity mostly come from the base to RNA interaction, and those base mostly in the loop region, because helix all look similar. Especially in viroid, it has short helix. It's not long helix. Okay, so now with all these spaces, we look at how viroid and RNA and protein interaction happens. So here, this is one of the RNA structure. It's 26, almost next to the terminal loop, this loop 26. And these 26, you can see a lot of arrows. Those arrows means you can modify those base by chemical reaction if they are not involved in base pairing. If they are free, you can label them. If they are in the base pairing, you cannot label them. Then you can tell a difference. And based on that, for example, this A, at one side is involved with U pairing, but still labeled with medium activity. But some of them has very weak, I forgot which one, I need to point out. But the bottom line is you can label the base with chemicals if they are not involved in base pairing and they have free, I think they have free sugar, then you can modify that sugar part, okay? And when the protein VRP1, Britton talked a lot about it, comes to this viroid region and it binds to it, and that forms a complex. Okay, and that complex recognized by another host factor, which is a general factor. They recognize nuclear localization signal, so they put the host into the through the nuclear pool and helps viroid get into the nucleus. That's the general model supported by some data. Okay, so the idea is when you have a specific motif, it gives a specificity to certain protein, so you can attract them to bind to you, and that gives the function to this complex. Okay. Another example I showed in this morning is the TF3A, and with the grains, the PSVD RNA genome, and this helps you to think the size of a protein. TF3 is 33 kD, 35 kD, around that, and PSVD molecular weight is 120 something kD, kilodalton. So protein is much smaller than the RNA, and you have that picture in your mind when you imagine what's happening in the cell. Okay, and we can map the binding site. There are chemical tools, and now we have prediction. Of course, prediction is not always reliable. You need to do some experiments to test it. And I have explained that. Another visual difference is here. We don't believe VRP1 changed the overall structure. You can model it, whatever, there's not much change. But when TF3 binds to it, it kind of bends it. That's a surprise to us. And this also gives you an idea. One RNA, same RNA, when they bind to different protein, the structure changes. And that structure changes often means function change to the complex. Okay? And this is more complex structure, but just emphasize again, TF3, in these slides, TF3 bent the RNA structure, so this part attached to part of RNA. When it comes to the PO2 complex, this part, the same part I just pointed out, now wandering around the polymerase, so anchor the RNA and protein to polymerase. Okay, so that part is kind of, how can I say, like a spring, can change the position. And the same RNA protein complex in different process, it has different conformation, looks like. Okay, so it's all dynamic, and now you can imagine why it's so hard to predict RNA structure. The same RNA, a different process, have completely different structure. And we don't have all the information yet. Allow us, like the protein, to predict that function. And that's why you need work on RNA to get more RNA data, structure data, and function data. And that's the key. Okay? Now here, RPL5 protein I didn't show you. RPL5 protein, we test, so this is called a gel shift assay. When you have a free RNA, of course, somehow we don't know why they give two bands, non-specific bands. Now, when we add the protein, the RNA shifted to the top. So because RNA protein has a larger molecular weight, so they move slower in the gel. Okay? If we use the wild-type RNA, we can see the shifting. It means the protein binds to wild-type RNA. And then we use a loop 4 mutant very close to TF3A binding site. It's not the RPL5 binding site. We can see the shifting like the wild type. If we use loop 26, which is close to VRP1 protein binding site, we still see the shift. It's the one in the middle, loop 15, also called the loop E. And that one, you don't see the shift. At that time, I was so excited. I was thinking, okay, loop E is the binding site. Now you see I have a question mark. Okay, and thanks to Darrell's work, make it really complicated loop E. So loop E can be written, if you read literature, can be drawn in different conformation. You can see this green part is loop E, and this part is also loop E, different presentation, quite different. Why that's the case? Okay, so he drew this thing. When the virus is replicating, they make multimer. You know what is multimer? Do you understand what is multimer? Okay, so see, everyone was talking about the multimer, tetramer, all the things showing that. What is exactly a multimer? Okay, you have a circle. That's a template, circular RNA. And the polymers sit on it, move, because the circle is not stopping. Keep moving. And the template, the product stick out. Then you got one round, that's one copy, not stopped. Another round, another copy, so that's dimer. You keep going. You can have 4 circles. You can get tetramer. Okay, and different viroids have different multiple copies. Okay, I don't remember it, but in the 1980s, amazingly, those scientists with that limited tools, they figured out HSVD has, I don't remember, 8 or 12, and PSV is mainly 6 copies in tomato, something. If you read all the literature, you can get all this information. The point I mentioned this is, when you have 2 copies, one copy and second copy, they are in the same molecule, one line, and then in the middle of it, they form this structure, okay? And this structure, you see they join here, like they attach. They are not, this is a kissing loop. You can think 2 pyramids now attached to each other like this, from distant side in 2 parts, okay? And that makes that loop region extended, stretched, become a double-stranded region. And that can be cleaved by, the reason it's called RNA3 type enzyme is because the character of the cleavage, the 2 nucleotides all hang and cleave here, and also the terminal structure, the hydrogen or 2,3 hydrogen or 3 hydrogen. Sorry, that's not my work. I didn't remember the details. And then, when it's cleaved, the rest parts, because of the self-complementation, fold back. Okay. It doesn't help me because I need something visual. So I draw it again in my own paper. So this is one copy in one molecule in magenta. And this is blue. And this is one copy, another copy. They all form this helping structure. And the kissing loop, they are distant. And when these 2 touch or kissed, now they stretch, become a double strand. And that being cleaved. And when it's cleaved, you will see this magenta hybrid with this blue. This is one copy. That's a unit copy. So they keep cleave, and they keep getting the monomer. That's how it works. We don't know the enzyme, but we know it's RNA3 type enzyme. Okay? And from this helping structure to here, loop E, then they can be ligated from linear form to circular form. That's how viroid does circularization. That's DNA ligase 1. They kept talking about DNA ligase 1. Okay? Now you will see here 2 different presentations of loop E region. And if I mutate loop E and see there's no binding, I don't know which part I messed up. So I cannot confidently now call it RPR5 binds loop E. Okay. So just give you some. So since this is science, okay, it's keep evolving the ideas, new data, keep you thinking deeper and deeper. This is the last slide. I make sure, okay, over time again. Sorry. This is the last slide. I will show you, this is not viroid related, okay, but I will show you why this RNA work is important. So I moved to Florida, and we have another faculty, horticulture science, a Canadian lady working on metabolite. Okay, she did unbiased genome screening, looking for some surpriser, and she mapped 2 mutations. Classic genetics mapped 2 mutations and the reward phenotype as she wants. But the mutation happened in 5' UTR. She had no clue how to explain it. If it's in the protein coding gene, you will think, oh, amino acid change. If it's in the promoter, you will say, well, enhanced transcription or reduced transcription, but it's in the 5' UTR. And I was there, I was trying to join the plant molecular cellular biology program to recruit students. And she introduced my work. Oh, you work on RNA structure? Okay, let's talk afterwards. And then she said, can you help me to look at this thing? I look at it. I cannot tell you the gene because this is her work and she's going through patent and publication, so I don't talk about the gene. Okay, but this gene, 5' UTR, has this kissing loop, and the kissing loop is very close to the AUG. This is AUG, the start codon for translation. And because that is a repressive structure to inhibit translation. And what she found is additional mutation at a different position. Okay, and that can release this kissing loop, make it open structure. Then what she got is translation enhanced plants always express the chemical she wants, and the plants look like normal wild type. So she can, and that's health benefiting metabolites. But this is a new way to think about it. You introduce single mutation to plant genome, and you change gene expression. And how many people are talking about enhanced crop traits? They're talking about, let's clone this gene, doing transgene. But the market doesn't accept it, transgene. Now you have gene editing tool. You know the RNA structure. You look at it, design it, and change that single base in genome. At least in US, it's acceptable, gene edited crop. Then you can get the crop on market directly. So that's from viroid RNA structure, so those basic blocks to agriculture in the future. Okay, so it means you study viroid like this generation, and if you want to explore more, that's also okay. There's lots of opportunity. Okay, so I finish here.